so now that we're officially done with Module 2, an overview, we'll now move on to Module 3. In Module 3, we'll be covering quite a bit. So we'll be going through effects of the environment on the organism, adaptations, the theory of evolution by natural selection, and evolution, the evidence. Okay, so 4.6 billion years in 1 hour. Yeah, there we go. Like, basically, this idea of all of the things that are involved at which times. So it's very interesting. Let's go through the peppered moth. So during the Industrial Revolution, the moth population changed from majority white to majority black. And a lot of people were like, whoa, how did this actually happen? And the theory is, well, what this is trying to say is that it was due to a change in their environment. So there was increased pollution and smog. So obviously during the Industrial Revolution, there was a lot of pollution, manufacturing, etc., and hence caused a lot of smog. So number one, within the moth population, there was always variations, as some were mutants born coloured black in a predominantly white population. All right, so you have some black and some white. So environmental selection pressure was applied to the population due to the change in environment because of the pollution. So birds could better spot white moths as prey and eat them than the black ones, which was then, camouflage. Most white moths were killed by predators, whilst the white moths actually survived because they were able to camouflage. As a result, moths with the black phenotype or physical structure reproduced and passed on those colourings to their offspring, and after many generations actually achieved the dominant trait. So that is sort of the diagram there as well. Okay. So biotic versus abiotic factors. So biotic factors relate to anything that's living, whether that be organisms living in the same ecosystem, competition for resources, so food, territory, predation, disease, etc. Whereas abiotic factors relate to anything that is non-living. So, for example, climate, temperature, wind, shelter, availability of food and energy sources, so access to water, light, essential nutrients, these are all non-living factors. Pollutants, chemicals, pesticides, insecticides, all of those things are non-living. So just understanding the difference between the two if you were asked that in an exam. So, cane toads, introduced to Australia. Cane toads were introduced from Hawaii to North Queensland in 1935 in an attempt to control populations of native beetles, grey-backed cane beetle, which ate sugarcane crops without using pesticides, because pesticides would also impact the crops, right? So spread. Cane toads started to spread rapidly, reaching the NSW border in 1978 and the Northern Territory border in 1984. The current population is estimated to be around 200 million approximately. This has resulted in the widespread ecological effects of the spread of disease, for example, the loss of biodiversity, spread facilitated by traveling on clear roads. So they would literally travel on clear roads all the way to these locations that were very far away. They were hitchhiking, building and landscaping materials, things like that. Evolution. So population has rapidly changed over the last 70 years due to special sorting. So toads naturally born with longer legs move further and they breed with outer toads who have also traveled far. So the genes for faster movement, when their genes are passed on, become concentrated at the forefront of the invasion, and they're passed on to the offspring who become really fast as well. This has allowed rapid evolution over 50 years. They used to travel 10 kilometers a year, and now 55 a year. So it's very, very, very fast, right? Compared to what it was. We can see that evolution occurring. Population changed. So cane toads on the front line of the invasion have longer legs, are more agile, have a reduced immune response as less energy is invested into it, in preference for consistent long distant movement. So cane toads can also rapidly acclimatize to colder climates by behavioral adaptations. So, for example, by basking. And rapid psychological shifts. So this will they have a lot of adaptations to adapt to different weathers, different climates, things like that, so that they can actually spread even further than what's anticipated, and they can travel very, very far. Next, we're going to conduct practical investigations individually or in teams, or use secondary sources to examine the adaptations of organisms that increase their ability to survive in the environment, including structural adaptation, physiological, and behavioral. So, adaptations. What is adaptation? It is simply a process by which an organism, or species becomes better suited to its environment. So, structural adaptation, for example, adaptations that form part of the physical features of an organism. So, physically, there's been a change. So, for example, rootless teeth which regrow in wombats. Physiological adaptations. So, adaptations which result in a chemical change rather than a physical change to the body of an organism. So, sunflowers grow towards light sources. That's an example of a physiological adaptation. Next up, we have behavioral adaptation, so changes to an organism's behavior in order for them to live in their environments better. So, kangaroos, for example, lick their forearms in hot weather. That's an example of behavioral adaptation. Structural, physiological, behavioral. Next up, let's investigate through secondary sources the observations and collections of data that were obtained by Charles Darwin to support the theory of evolution by natural selection. For example, finches in the Galapagos Islands, as well as Australian flora and fauna. Know these two examples, they're in the syllabus, which means you need to know them. So we'll go through them. So Charles Darwin was an English naturalist, developed the theory of evolution by natural selection. So where's his voyage? You can see it sort of goes through Australia, went through a bunch of other places as well. So Charles Darwin joined other scientists on the HMS Beagle to travel around the world and collect natural history and geological samples to try and prove his theory of evolution. Okay, introduction to the theory of evolution by natural selection. So, change in a species over many generations as a result of natural selection of favorable characteristics is this theory. Evolution is a gradual process. It cannot happen overnight. It happens over many, many years, and it's facilitated by the mechanism of natural selection. So, steps of evolution. How would this have occurred? First of all, there's a variation that occurs within the population due to natural mutation. So think back to a variation of the moth, so that one was black, or there were a few black ones and a few white ones. Selection pressures are applied, so physical change, chemical change, competition, so changes in biological niches are adapted or inhibited. Sorry, the second one should be where we say black or white moth. Phenotypes are best suited to the environment survive. So think back to, okay, the black moths were more likely to survive because they could better camouflage, and the white moths couldn't because the birds would eat them. Or in this sense, certain finches will develop suitable beaks and consequently survive as they can still eat. Surviving organisms then reproduce, and then they pass on those traits, which is a very important point. They pass on that trait to the majority of the population. What is a selection pressure? I've been saying this term, but if you're unsure of what it is, it's an external agent which affects an organism's ability to survive in a given environment. So, investigate through secondary sources the observation collection of data. So, we'll go through finches of Galapagos Islands and then explain using examples how Darwin and Wallace's theory of evolution by natural selection accounts for convergent and divergent evolution. So, we'll go through that now. So, different evolutionary processes. So, convergent evolution, this is the process by which organisms which are not closely related evolve to have similar traits because they are adapted to a similar environment. So, similar environments means that they have similar suitable traits, right? So, they sort of essentially grow to have similar traits because they're all in a similar environment. Whereas divergent evolution is a process by which organisms which have recent common ancestors, so they're closely related, evolve to have different traits, right? So that's a difference because they have adapted to different environments. So they have a common ancestor, explained by Darwin's theory through the example of the finches from the Galapagos Islands. So, why Darwin's finches carry the entire biology syllabus? Let's go through it. So, why Darwin's finches is better than every other case study ever? Because there's just so much information provided. So, the evidence. So, Darwin's finches are an example of gradual speciation. There are 14 species diverged from the common ancestor of the mainland finch. Okay, mainland finch. Darwin discovered 12 of these species, which had never been discovered before. Finches developed different beaks sizes and shapes to suit the diets in their individual biological niches. So we've got some fruit eaters, insect eaters, we've got some seed eaters. Their beaks were sort of shaped accordingly. Why the finches make the biology course? They actually demonstrate how species adapt as a result of selection pressures, which is something we went through earlier, the external agent, remember? External agents which affect an organism's ability to survive in a given environment. Remember that definition? That's why it's here. They're a great example which can be used for multiple points in the syllabus, which is why I would recommend making sure you do know this example. So, Darwin, Australian flora and fauna. So, instead, this was an example of convergent evolution, where unrelated animals adopt similar features due to similar selection pressures. For example, Darwin noted that the Australian platypus behaved in a similar fashion to the European water rat. Okay, next up, analyze how an accumulation of microevolutionary changes can drive evolutionary changes and speciation over time. For example, the evolution of the platypus. So, platypuses are believed to have diverged from the following lineages. So, we've got due to a blend of characteristics. So, we've got platypus, we've got reptiles which have venom, birds which are egg laying, and mammals which have lactation features. All of these are present in the platypus. Evolutionary change. So, this refers to the genetic change in species over time in response to different environmental selection pressures. So, this change is due to the process of natural selection, which involves Darwin's 4 points. So, these 4 points are proposed by Darwin. So, we've got microevolutionary change. So, evolutionary change within a species or small group of organisms, especially when it's over a short period of time. So, that's why it's called micro, because it's a small group or a short period of time. Macro is looking at a combination of microevolutionary changes which can cause speciation, so the formation of an actual entire new species. Macroevolutionary change. The platypus. Species are a classification of a group of closely related organisms which possess very similar characteristics, who can breed and create fertile offspring. That is a species. So let's go through macroevolutionary change. Another example is of the horse. So we're looking at the analysis of how an accumulation of microevolutionary changes can drive evolutionary changes and speciation over time. So, for example, looking at the evolution of the horse and the platypus, this one's the horse. So you can see 50 million years ago versus 35 versus 26 versus 3 million, and how it's actually changed. How the forefoot and the skull has changed over time, and the length of it as well, going from 38 centimeters to 52 centimeters to 100 centimeters to 135 centimeters. So, the first type, the dawn horse, it's likened to a dog rather than a horse. So, what we would consider a dog today, kind of like a dog shape or whatever, is what that was. 4 toes on each front foot and 3 toes on each back foot. And they have omnivorous teeth. About 30 centimeters tall. And you can see the forefoot there. Then we have the next type, which had more grinding teeth, and it was to suit a plant-based diet. Okay. Next up, we have the next type of horse, which had even tougher grinding teeth and larger with longer legs, aiding with speed in open areas. So we can see that they've got the forefoot there as well, and the difference between the third to the second and the first. Finally, we've got the last type, and they've got hypostodont teeth. So teeth which grew continuously out of the gums as they were worn out. So developed jaws with ridges for efficient grinding, and long legs and body size aided in running in open areas. So those are the 4 different, steps, and you can see those steps of how the horse has changed over time to create macroevolutionary change of the horse. So the equus, or the modern horse, looks at this idea of the horses beginning to evolve with singular toes, strengthening their toe ligaments, and removing the side toes, which may not be as much used. So they're not really using them as much. So they're trying to. So over time, this sort of disappear. Appeared around 4 million years ago and is what we identify as the modern horse that we see today. What are the features? Long legs, long necks, horse-like body, deep jaw, and long faces. So that is the macroevolutionary change of a horse, and then we saw the macroevolutionary change of the platypus as well. So this idea of lineages and the different characteristics being developed and put into that new fertile animal. And we saw it with the horse. Now we're going to move on to punctuated equilibrium. So, explain how punctuated equilibrium is different from the gradual process of natural selection. Let's do it. Until the mid-20th century, evolution was considered to only occur by the gradualism Darwin suggested, with slow changes in a species over time due to natural selection. Alright, so this idea that it was a slow change, right? However, in 1972, Eldridge and Gould instead suggested that evolution is far less consistent and gradual as Darwin theorized. Punctuated equilibrium is believed to be a period of rapid evolutionary change, leading to the emergence of a new species, followed by a period of stability and stasis where the population remains unchanged. So gradualism, we can see here. Whereas punctuated equilibrium, we've got the changes, and then for a long time, they're sort of stable, they stay as they are, and they're not changed. So just understanding the difference between gradualism versus punctuated equilibrium. So, punctuated equilibrium, we can see the evolution in time and we compare that to the theory of evolution. So, theory of evolution says new species formed, new species formed, new species formed over time, like it's as time goes, like it is just new species being formed. Whereas punctuated equilibrium says, wait a minute, that's not exactly how it works. It's this idea of we have evolution, we have time, we have a new species, but there is stability in that process and we're sort of stable for a while before we see a new species again, and then again we're stable for a while, and then we see a new species, we're stable for a while, and that process continues. Rather than just new species, new species, new species, punctuated equilibrium takes a more conservative approach and says, actually, no, let's calm down, let's have a period of stability again, then we have a new species a little bit later. Okay, so the theory of evolution is sort of go, 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 whereas punctuated equilibrium sort of goes calm down. Okay, let's sit down, let's take a break. Okay. Moving on to amino acid sequencing. So, where is this in the syllabus? Investigate using secondary sources evidence in support of the Darwin and Wallace theory of evolution by natural selection, including but not limited to biochemical evidence, comparative anatomy, comparative embryology, and biogeography. So, amino acid sequencing, two organisms sharing part of their amino acid sequence, is strong evidence for divergent evolution. Going back to divergent evolution, remember what we went through over here. Remember the process by which organisms that have recent common ancestors but they have adapted to different traits in different environments. The horse, yeah, it's fine. Here. So amino acids are the building blocks of proteins. So it's a really great way to figure out if these, like to look at the amino acids and compare and see if the divergent evolution has actually occurred. So how does this actually work? Because it doesn't sound, it sounds really like quite a lot to actually go in and see it. And you can see in the photo, it doesn't look quite as simple as we thought. Scientists compare amino acid sequences which make up specific proteins of different organisms. For example, the cytochrome C, the hemoglobin, etc. So we can see here the phylogeny based on nucleotide differences in the gene for cytochrome C. So humans, monkey, donkey, horse, dog, horse, donkey, pig, rabbit, kangaroo are sort of having a similar wavelength. And then we've got the pigeon, duck, chicken, penguin, turtle, all in tuna, all in the same sort of wavelength. And then we've also got like a bunch of other ones there as well. Organisms which share part of their amino acid sequence. So we've got molecular homology of cytochrome C. So amino acid position 1, 2, 3, whatever. And we've got the human, chimpanzee, pig, chicken, dogfish, wheat, yeast, etc. And we can see that. Right, so the source is from the Year 11 Biology in Focus. Now moving on to biochemical evidence looking at DNA and DNA hybridization, which allows us to determine relatedness through comparing nucleotide base pair order of different species. What is this process? So we've got the double helices of the DNA from two different species, which are split when you've got the 1994 heat applied. The separated segments of DNA from the two species are then mixed. You can see here, got the species A, species B, then they are mixed together. These mixed strands then combine to form a hybrid DNA molecule in a process called reassociation. Closely matched base pairs will strongly bind to one another. Okay? So if they strongly pair to one another, heat is applied to separate the strands once again. The higher the temperature, the closer the bond. The more nucleotide base pairings which are closely bonded means that they are more related they are as a species. So we can see over here sequences similar and they are very difficult to separate, right? Because they're all connected, you can see, compared to this one which is not connecting. This means that species A and C are closely related. compared to species A and B, where you can see that they are actually not connecting and they're dissimilar and they're very easy to separate, meaning that they're not closely related. Moving on to comparative anatomy. Comparative anatomy is the study of similarities and differences in the structure and anatomy, essentially, of living organisms. So, key terms: homologous structure, structures which are the same or similar, homology, same, right? So, but due to modifications through the course of evolution, may have or serve a different purpose or function than what another organism has the same feature or has for their function. Pentadactyl limb is a five-fingered toed limb in vertebrates, which can be compared to evolutionary relationships. So 5 fingers in humans, and we can see that also in horses. We can see that in the whale, the turtle, the frog, the bird, but very different uses, right? So the bird will fly, the frog will hop, turtle kind of waddles, I guess. Horses run, humans use our limbs also for walking, but also for hands, picking up things, etc. So it supports the ideas that species have evolved from a common ancestor and therefore share similar features, but also that due to divergent evolution, there are particular modifications between each species. Noting that this is a theory. Okay, so this is a theory. It is not based, I guess, on like this is an idea that has come about, and this is an evidence to support that. Comparative embryology is another sort of evidence, or I guess way that they are trying to prove this idea of theory of evolution. So this comparison of the developmental stages of different species, right? So similarities in early development of organisms is strong evidence for divergent evolution. Why? Because it highlights the similar features and structures. So, for example, we can see the slits being seen in the early developmental stages of fish, reptiles, birds, and humans. So, however, these develop into very different features. Okay, so we can see here fish, salamander, tortoise. You can see how different they are to the human. We've seen the rabbit, the calf, the pig. Very different, right? But we can see those essential similar features and structures when we first start off with the embryo. We can see here as well: fish, reptile, bird, human, and the slits. Next up, biogeography. So again, we're looking at biogeography. Biogeography, what is this? It's the study of the geographical distribution of living and extinct organisms. So, to support Darwin's theory, he sort of said that new species evolve, right, from a common ancestor when geographically separated from the rest. This is an example of divergent evolution. This can be illustrated through the example of ratites, a group of flightless birds found all over the world who were either isolated when the continents separated, or who flew to their respective continents before developing flightless features. So kiwi in New Zealand, the emu in Australia, the cassowary in PNG, ostrich in Africa, and the rhea in South America. All sort of flightless birds. But they said that in accordance to Darwin's theory, he said, well, that's because of this. He said that essentially what I went through, right? So this idea that they flew to their respective continent and then they developed flightless features, whereas once they didn't have them. So, relative dating. So, this is a form of dating which relies on the assumption that older fossils will be in lower layers of sedimentary rock and younger fossils will be on upper levels or layers as seen in the diagram. I'll show you in a second. So, it does not allow us to determine the exact age of a fossil. Sorry. So, chemical analysis, looking at this, right? So, chemical analysis looks at measuring the amount of a certain chemical, such as uranium, fluorine, in the bones, and nitrogen levels can also be monitored. Stratigraphy, so this idea that this previous concept, which is reliant on sedimentary rock forming in layers which contain the oldest fossils right down the bottom. And biostratigraphy is this idea where fossils, which are known to have existed in a certain period, will be compared to isolated fossils to help determine their age. So there's this sort of trying to figure out what age they're at the moment. So absolute dating, it just enables the age of the specimen to be determined using radioactive elements within the specimen. So it provides us with a quantitative age for a fossil rather than just aging it in relation to other fossils, which we saw over here in specifically biostratigraphy. So fission track dating, so allows us to date a fossil looking at its uranium levels over time. This element can split in a process called fission. Right? So marks called fission tracks can form due to atoms moving at high speeds throughout the substances during this division process. So the number of these fission tracks allow us to identify how much uranium is present and therefore how old the fossil is. So this is used to date fossilized teeth between 5,000 and 100 million years old. Whereas when we compare that to radiocarbon dating, for fossils less than 60,000 years old, the known rate of decay of a radioactive element can be compared to the amount of carbon within the sample fossil. So, for example, the quantity of the isotope carbon-14 can be measured in a fossil to estimate how long the element has been present within the fossil, and then we can go from there and conduct radiocarbon dating. That concludes Module 3 and the sort of summary of Module 3 and the areas consisting around that. Hopefully that was beneficial to you and you sort of understand more clearly what's involved in Module 3 and some of the concepts that are there. We're now going